Good morning, everyone. My name is Gaoxiang Zhou. I'm here to present my work, Intervention Pathway Discovery in Complex System Scenario-Dependent Sensitivity Analysis. It has been proved that sensitivity analysis can help identify and explain the influence of external and internal changes on the model and its components. So we here propose a comprehensive framework to study the sensitivity of the biological network and how to identify key pathways to target interventions. First of all, I want to introduce the model approach we use to test the framework because we all know there are a lot of biological modeling approaches. Within our element-based models, the whole population of the same component is a unit, which we call an element. The modeling approach is capable of integrating knowledge, handling feedback, having less complexity but more information. Technically, for each element, we define its update function by summarizing the relationships of all regulators. For example, figure 1a is a model of all Boolean elements. Secondly, an update scheme is required for the model to be executable. So we apply a random scheme in which only one random element is allowed to update at each time step. With that in mind, the main focus of this work will be a variety of sensitivity analysis, such as immediate and remote sensitivity, sensitivity under uniform and scenario-dependent distribution, Boolean and discrete sensitivity, as well as the application in pathway discovery and how we are going to validate it. The roadmap of our analysis framework is quite clear. That is, we first construct a hybrid model, define scenarios and run simulations. Then, for each directed collected element pair, we calculate the influence. Hold on, I will share you the calculation details momentarily. Next, we can add those influence-based weights to the biological networks to be a weighted graph. Finally, we shall be able to conduct many tasks with this graph, such as comparing scenarios, finding the most influential elements or pathways. Now we have to face the fundamental question. How is influence calculated between two elements? Intuitively, the influence is high when one element change always leads to another element change. Therefore, the immediate influence of one regulator xi on x is obtained first to enumerate all xi's possible change, substitute them into update functions, and find the average change in x. That's it. When we take all model joint state into account, we can have sensitivity under uniform or scenario-dependent distributions. Also, if we add with wij being lective log of rij to the edge from xi to xj, we can also explore many pathways, with each pathway assigned a score calculated from those weights. The larger alpha is, the more influential the edge is. Accordingly, the less the weight is. So the most influential path is the one with the minimum weights. Without further talking, let's move to a case study where we apply the framework to the T-cell differential model of 55 elements. We show in figure 4a a weighted directed graph of the T-cells model with some key elements. Then figure 4b gives the sensitivity values of all elements. Some of them, for example, RAS, has relatively higher sensitivity under uniform analysis, while others like PKC, theta, and IL2R are more influential in scenario analysis. Generally, remote pairs are difficult to detect in a large model. The element-to-element influence matrix in figure 4c clearly shows which remote element pair is more sensitive and correlated to each other. This can largely narrow down the intervention candidates. We also apply the pathway discovery to the T-cell model to extract the most influential pathways targeting FOXP3, the dominant components in the T-cell model. We don't stop here and take further steps to verify our extractions. That is, we remove the discovered pathways from the model and test whether the model properties are preserved compared to some randomly removed pathways. Figure 4D shows the significant changes in property satisfiability. Both uniform analysis and scenario-dependent analysis achieve such a goal, while the latter even performs better. We also do cross-scenario verifications in property satisfiability. That is, to remove based on scenario A, but test on scenario B. Figure 4E shows the model is diminished only when the removal and testing are based on the same scenario, indicating the value and uniqueness of a scenario-dependent study of sensitivity. Overall, adding sensitivity-based weight to the network help identify important pathways, suggest the most suitable interventions, of which scenario-dependent study take the context into consideration, shows even great power in those applications. That concludes my presentation today. Thank you.